Hi, my name is Dan Rokhsar. I'm a professor at UC Berkeley, and I also work at the Department of Energy Joint Genome Institute. My lab works on lots of plant and animal genomes, and really happy to be an early adopter of Dovetail technology. The project I want to tell you about is cassava. Cassava is a tuberous crop. It was originally domesticated in the Amazon basin about 6,000 years ago. But it was brought to Africa in the 1500s and 1600s by Portuguese slave traders to feed the people they were going to kidnap. And it was established in Africa and other tropical regions around the world, and now, 500 years later, feeds about 500 million people. So in this project, we work with scientists from Africa and around the US and Australia to generate a reference genome for cassava, and then to integrate it with all the breeding information. So we had several goals. One, we wanted to make a chromosome scale sequence that we could integrate with genetic maps to enable genome-assisted breeding approaches. We're also interested in the genetic diversity in Africa because since it was originally domesticated and is native to South America, there was some concern that the germplasm diversity in Africa would be too low. And so we really wanted to have chromosome scale sequence to really look at this from a whole genome perspective. The cassava genome is not particularly large. It's about 750 million bases long, but it's pretty highly repetitive. And it's also a paleotetraploid, meaning that most genes are found in 2 copies. And so those were some of the challenges we faced in sequencing the genome. So we sequenced the genome using an Illumina method, and then we added Chicago library sequences using the high-rise assembly method, and it worked spectacularly well. Even though the genome is repetitive, we were able to organize the sequence through these repetitive regions, and by integrating with a dense genetic map, we were able to obtain entire chromosomes. So the dense genetic map allowed us to do 2 things. First, it confirmed that the high-rise assemblies were very accurate. And second, it allowed us to assemble all 19 chromosomes, and we were able to place 97% of the genes on those chromosomes. We have a very complete and well-annotated sequence that now we could take a look at. We were fascinated to find that many of the African cassavas had chromosomal segments in them that were from India rubber. Now, India rubber turns out to be a related species that was also grown in Africa by colonialists, but is not susceptible to some of the diseases that cassava is susceptible to. So by crossing India rubber with cassava, people were able to create more disease-resistant varieties. And by having the chromosome scale sequences, thanks to Chicago and high-rise and the genetic map, we were able to show that these were large chunks of sequence that were introgressed and understand how that happened. So now, thanks to this chromosome scale analysis, we have identified large chunks of genome that are associated with disease resistance, and we're following up with our collaborators to really understand what genes or gene variants might be associated with that resistance. We also found that if you looked in South American cultivars, you could find other short segments that seem to be from other species. And this is really informing our understanding of how to think about cassava as a species relative to all the other members of its genus. You can read more about this in our Nature Biotechnology paper that was published last May 2016. We're very excited about Dovetail and are now using Chicago and high-rise on several other plant and animal genomes.